record to the cloud. So, couple of housekeeping details. Some people have been, again, watching the Sapling Plus stuff. Sapling 4 was due this week. I got a couple questions about this. Please remember to read Piazza because the due dates are always listed in Piazza. But also, take a deep breath. I know that we're all struggling with the online formats, whether we're in synchronous or asynchronous meetings. It really doesn't matter. Everybody is struggling through this, and I include all the professors. I am in my office on campus on Tuesday and Thursday mornings. So if any of you want to either drop by, chat about stuff, you want to do a Zoom meeting, just send me a quick email. And, we could do that. That's no problem at all for me. I'm going to move this a little bit here. So you may have noticed I'm in a different spot. I'm on Magic Long Beach Island. Of course, the day I decided to come down here, it's raining. But at any rate, so just remember that flexibility is our middle name this year. That's what the F in GFB stands for, flexibility, okay? And so we want to make sure, I just want to make sure that this is not saying wait until December 11th to submit all your assignments, all right? What this is saying is take a deep breath and realize if you're a day late or 2 days late or, you have to work over the weekend, however it works out, it'll be fine. It'll be fine. So take a deep breath. Don't panic. I know a lot of you have high expectations for your future. You're already thinking about applying to professional schools. Everybody wants good grades, and you're all probably type A personalities and maybe some type AA or even an A personality, and it's okay. And I just want you to know, I'm familiar with the type. I've been there myself. And, but sometimes, and especially in these unique circumstances, you just need to take a deep breath. So if you're worried about chapter 4 and you can't get it done today, it's okay. Get it done over the weekend. Everything's fine. Next week on Monday, you'll see that chapter 6, we're rolling through chapter 6. And basically, there are a couple of readings coming up. Remember, you should be talking, I forget the chapter, but on multiple alleles and pedigree analysis. And the pedigree chapter does a little bit on genetic testing. And I want you to keep some of those ideas in mind as you go through chapter 6 in genetic testing, because we're going to cover a little bit more detail later on in the semester once we get through biochemistry of the gene and can talk about how contemporary, what used to be called next-gen sequencing, next-generation DNA sequencing, has influenced what we know about genetic testing today, ranging from personal genomics to actual clinical testing. So, you've got to understand the basic genetics first, then understand the biochemistry of the gene, and then we'll be able to talk about how applications of contemporary genetic ideas integrate, how the biochemistry integrates with traditional genetics. So we're right at that spot right now where we're getting ready to end the traditional genetics portion of the course and move on to the biochemistry of the gene. Some of you are going to be much more comfortable with the biochemistry of the gene part because there's less math, okay? There's only 4 bases. They are complementary paired. There's really just a lot less math, and you don't have to worry so much about it. However, it does present other kinds of problems because there's a level of chemistry, and in particular organic chemistry, that you need to know in order to move forward. And that's why organic is kind of a co-rec here. Do I think you'll be in trouble understanding it? if you haven't finished or completed organic yet? And the answer is no. I don't think you're going to have a problem. And especially with me, because I try to keep the molecular biology in this course at a sophomore level. I know that a couple of the other professors really ratcheted up and into a junior or senior level course, sometimes assuming that you've actually had not just organic, but biochemistry. I don't do that. Again, it's because I'm old. You can see my white beard now shows up very nicely on screen. And you know, when I was going through my biology curriculum, biochemistry was not an undergraduate degree, or an undergraduate course rather. Biochemistry was a graduate course, and in fact, the first year of graduate school, I had to take two full semesters of biochemistry, and it was pretty much my worst nightmare. And little did I know that I would end up doing 2 postdocs in biochemistry, and actually became really good at it. But one of my postdoc advisors, who is head of chemistry at Rutgers, this was my first postdoc at Rutgers. I was actually assigned 50% of my time as a postdoc to the Department of Chemistry, and we were doing protein sequencing in the lab, and it was great work. I love being a protein chemist even more than I like being a DNA chemist. DNA chemistry is easy, protein chemistry is hard, just remember that. And I remember my advisor saying, well, you covered this in your biochemistry course. And I was like, oh no, I never saw this chemistry before. Who knew any of this anytime? I never did it. He said, do you have your biochemistry book in your office? And I said, yeah. He said, go get it. So I went it out. He said, turn to chapter 16, or whatever chapter it was. And I said, okay. So I turned to chapter 16, and chapter 16 is there in what was originally my brand new book, with all this yellow and pink highlighter and my handwriting and notes in the margin. And he said, see, you did it. And I was like, I don't remember any of it. So just to let you know, I really wouldn't force that on anybody here. But there are things you need to know to get to the third section of the course, which is really applications of contemporary molecular genetics and how it applies to heredity and patterns of inheritance. So that kind of gives you a sense of where we are. If you might have guessed, because we are finishing the first third of the course, we're really on target to have our first exam on the 9th, which is next Friday. So exam 1 will be next Friday. Exam 1 will consist predominantly of problems. All very, very similar to the ones that you've been doing up until this time. I plan on taking about half the questions from Sapling Plus and a couple from probably the problem set from last week, and then of course the problems, some of the problems that we'll talk about today. And I will actually, because next week are the pedigree chapter, remember there'll be at least one pedigree on the exam. Shouldn't be awful terrible. I'm trying to figure out the presentation. I think we're just going to do it on Blackboard, I think is the best way to do it. And it'll be all set up for Blackboard. It will be timed so that there will be, basically it's what you can accomplish in the context of a full class period. Okay, so an hour and 15 minutes, basically 75 minutes. People who've got any kind of extra time they need or whatever, we'll work that out, and it's real easy to do in Blackboard, so don't worry about that. Other than that, it's really, it ought to be really straightforward. It ought to be really straightforward. The second exam, just so you know, the second exam will be nothing like the first exam. The second exam, which will cover all of the molecular biology, or at least what I prefer to call the biochemistry of the gene, will really be all multiple choice. And so little bit different perspective on the two different areas of genetics that we'll cover in the first 2/3 of the course.
I have two questions.
Excellent. Let's start with number one.
So for the my first question is it going to be posted at like 8:30? Just because I have like a class like 10 minutes after this in person, so like I'd be doing the exam in my car. So I'm just like wondering like.
No. I'm sorry. It'll be available for the entire weekend.
Oh, okay. So you can.
But it will be timed in the sense that once you start, you got 75 minutes to do it.
Okay.
Unless you have a different timing schedule. But other than that, no problem at all.
And then my second question is, so basically it's like half like problems and like half multiple choice, like from the Sapling Plus? Or is
They will be, well, I hate to call them multiple choice. It will involve you solving a problem, but the answer may be, given in a series of, potential answers. Yeah.
Okay, cool. Gotcha.
But it won't be like, true, false, or.
Well, yeah, gotcha.
That's boring. Okay. So I wanted to take a look at a couple of things today, and basically all of the problems that I'm going to ask you to do are actually, let me see if I can find my screen here, are actually in that first problem set, and I'll show you those in just a minute. But I wanted to take a chance to remind you of how complicated things can get. And I think that that's really, to me, it's one of the fun things about genetics. And I'm a huge dog lover. And in fact, my dog is sleeping at my feet right now and not letting me move from this chair. I think she misses me. When we think about dog colors, there are really two main loci. And by the way, this can be said of most vertebrates. There are two main loci that control both skin color and, in fact, hair or coat color, pelage from our guinea pig example. And these are the two loci, B and E. And the B locus was originally named B because it was black, and black was dominant to brown. Later on, as the chemistry moved forward, we realized that these were actually two types of melanin. The black is actually melanin. Brown is something called eumelanin, and EU is one of those great prefixes from Latin in English that's used very commonly in scientific terminology. And the prefix EU means true. You guys know another word from Bio 1 that uses the prefix U?
Eukaryote.
Yeah, eukaryote. So what's karyote? Which is really weird because EU is Latin and karyote is Greek. So it's kind of a weird combination. But what's karyote? Oh, just shout out. I can't read my chat right now.
Nucleus?
Yeah, nucleus. Absolutely. Chromosome, nucleus. So eukaryote is true nucleus. Prokaryote is before the nucleus, right? Because prokaryotes don't have a defined nucleus. So, and actually we will come a little bit later on, we will come up with another couple of examples where in genetic terminology, certainly, we use the EU prefix. So melanin is black, eumelanin is brown. black is dominant to brown. And of course, because of the work on the genome projects that have been done over the years, and because of some great biochemistry, we now know that what the B locus is actually a tyrosinase protease. So tyrosinase, again, get used to the etymology of these words. Tyrosine, okay? The suffix ase, everybody knows, immediately means a what? Go ahead and shout it out. I know more than most of you know this.
Like a protein
Enzyme.
or an enzyme?
If something ends in an ace,
Is it like a protein or
Enzyme.
or
it's
something?
exactly an enzyme. That's right. Proteases, DNases, RNases, we'll talk about later on. This is a tyrosinase, and tyrosinase, of course, is an enzyme that acts at tyrosine. And the amino acid tyrosine is a precursor to melanin, So this is actually a facilitator protein that helps tyrosinase work to produce the maximum amount of melanin. You actually, as you've been working in lab on GeneVenture, have actually seen the B locus in action in the little molecular, or rather the cellular diagram of how melanin is put together by two major protein subunits, and then with a connector protein in between. And what the connector, as you increase the connector protein, you get an increase in production of melanin. And in that case, our dragons turned from orange to gray, or even you could even get darker according to the model that they set up there. So you really can get this idea of the metabolism of tyrosine as it relates to melanin and pigmentation is extremely important. So this does not inhibit the enzyme completely, because if it did inhibit the enzyme completely, there would be no dark color at all. There would be neither melanin nor eumelanin. The other, and by the way, this discussion of melanin fits in really, really nicely with a new video that's going to be about the evolution of human skin color by a colleague of mine from Penn State. Both she and her husband are just spectacular scientists, and really excited to share it with you so you could see some of the ideas behind what really contributed to the tremendous variation in skin coloration in human beings. And also should be very, very clear in beginning to address this concept of race, which is really, as most of you know, race is a social construct. It is not a biological one, and you'll see the reason for the different groups of people who have different geographic origins and how their skin colors have changed over the generations. So really great video, really easy to watch, a lot of fun, and I think you'll enjoy it. So that'll be up on Piazza this afternoon, so you should be able to get a good look at it. The other locus that influences coat color, of course, is the E locus, and this is the melanocortin receptor. And if you want to look up the gene numbers, that's what those abbreviations are. You can actually look these up on any of the genome projects. And in fact, I'll show you how to do that a little bit later in the semester. And the E locus is one of those cool loci. The E locus is what's considered to be a permissive locus. In chickens, by the way, the exact same gene is called the C locus. It doesn't stand for chicken, it stands for color, by the way. And it's a permissive locus, and that is, it has to be present. So in other words, and think about this from a biochemical point of view, it is the melanocortin receptor, okay? So if the receptor is there, it reacts to all of these other products and enables the biochemical process to move forward. If there is no receptor, it doesn't matter what all the other biochemical components are and whether or not those pathways exist. If there's no receptor, there's nothing to turn the system on. Okay? So it is a permissive gene in the sense that if it exists, you get color. If you have a mutation in it where you have a non-functional receptor, you get no color. So the ones we tend to think about are CAPI and small e, but I do want to make things a little more complicated and just remind you a little bit that there are actually multiple alleles where the receptor can be overactive, which would be the E superscript M allele, or it can be underactive, meaning it still exists but does not function at the same level as the wild type allele, and that's the EG. So there are at least 4 alleles that we know of at this particular locus. Now, hopefully some of you in your heads are thinking, wow, do I have to do a Punnett square with all these different combinations? And the answer is no, you don't. But I do want you to just be thinking about how complex some of these patterns are and how a relatively simple system of a locus with 2 alleles can produce a huge range of phenotypes, okay? So if the animal is homozygous small e, then a compound called pheomelanin is produced, and pheomelanin is basically a precursor to melanin and or eumelanin, all right? So it's a precursor. So you only see the precursor, you don't see the final product. And these are dogs that are red or yellow. So let's see how we're doing this. Oh, it's not, there we go. So if we want to think about our labs, and this was a problem I used in an older, one second. There we go. This was an example that I used in an older set of problems and presentations. And if you think about your labs, anybody got labs at home? Yellow labs, very, very popular. Okay, right? And if you have a yellow lab, you know, oops, don't panic. There we go. Oh, come on. There we go. If you have a yellow lab, you know that the yellow lab is homozygous. Same thing for a golden retriever, very popular dogs. You know that they are homozygous at the E locus for the homozygous recessive allele. And so these are the yellow dogs. Red dogs, same thing. If Irish setters, anything that's in the red variety are all homozygous small e. So whereas if they are black, they are of a different genotype. So you can think about, let me change the color of my pen here, somewhere. This computer is not nearly as good as the other one when it comes to changing colors. Okay. I just want to change my ink color. I don't think you could see the black very well there. There we go. Okay, so if you want to think about all the different possible genotypes that you might be able to put together, so you could see that any black lab will be cap B and either small b or cap B. and we'll have to have the permissive locus, and doesn't matter what the other one is. So this would be our black lab, okay? If you have a yellow lab, all right, what does a yellow lab have to be?
Homozygous recessive.
Exactly, homozygous recessive. What about at the B locus, though? It doesn't matter. Doesn't matter because the E locus is permissive, okay? So it's one of those things. So now you can kind of get a sense that, and you don't have to do this particular problem. This is just to get you thinking. You could think about all the different ways depending upon what kind of a lab you wanted to produce, okay? And what's the other kind of lab? Black, yellow, and?
Brown.
Chocolate.
Yeah, chocolate or brown, excellent. So if we were that, because we have to have the permissive E locus, but we have homozygous B, small b, so eumelanin's produced, and here are our chocolate or brown labs. So shows you really kind of a unique combination of genes can create this wide range of color variation. I should point out, just for honesty's sake, and to let you know, it's a hell of a lot more complex than this, even this example, in that there are more than just two loci involved in color or coat color in dogs or humans or anything else for that matter. So, but it is fun to look at it. There are a lot of modifier genes that can be redder, that can be a little bit different, a little more yellow. And of course, there are genes, notice that the EE is permissive for color, but notice there's no such thing as a white lab, okay? There is no such thing. There are white dogs, right? I'm sure you've seen them. There are white dogs, but they are really not at either the B or E locus, and there are actually two types of white dogs. One is actually white, and the other is albino. And the white is another one of those permissive color genes. It's actually, and actually it's not permissive. I apologize, I misspoke. It is actually an inhibitor gene that inhibits all coat color. What's the difference? How can you tell the difference between a white and albino?
Their eyes.
Yeah, absolutely. Eye color. Eye color. And I'm sorry if anybody's answering by chat. I can't get my chat screen open on this computer that I'm using here. So what color would the eyes be in an albino animal?
Red.
Right, red or pink. How about in a white?
Black,
Non-albino white.
black or brown or something?
Yeah, it would be colored. It would have color, black, brown, whatever color the eye is. So we do see these different types. And in fact, in lab next week, we'll show you some examples of these. Next week, we're doing some genetic variation in animal populations next week. And we have a nice slide set put together. Usually next week in lab, you guys would be in lab and we would have all sorts of animals set up for you. Fish, frogs, mice, and snakes, which are great fun. And usually One of my favorite parts. Snakes are another one of my favorite animals. They're just like pre-chickens, so you got to kind of figure it out like that. And it's great fun because for a lot of students, it's their first opportunity to ever actually hold a snake. And so I think that that's great. And some people continue to be a little bit afraid of reptiles, especially snakes. They got a bad rep, I mean, from the beginning when they tried to get Eve to eat the pomegranate or whatever they gave her, it was just they just get a bad rep. They're not slimy. They can't bite their tail and roll down a hill and chase you. They can't, they're really more afraid of you than anything else. But so we'll at least see some great pictures. And maybe if we ever get over all this COVID nonsense, you all have my personal invitation to show up at my genetics lab when we do this, and we'll hang out and we'll play with the animals, which is really one of the great things you can do. To kind of tie all this together, and the reason for this is as you begin thinking about using Punnett squares to create all these different types of animals, okay? The black locust, the E locust, all right? The white locust, or the inhibitor of color in skin and or hair or pelage. The albino locust, which eliminates all color. You could see that we're accumulating loci and we're accumulating number of alleles, and things just get very, very complicated, even with just a handful of alleles. And I want to show you how, if I could find my thing here, there we go. Let's get rid of all this crap, because I got a slide coming on. Go away, go away, go away, go away, go away, go away. This is kind of fun to do if you ever get a chance to play with these things. That was fun. So what I'd like to show you is just how complicated things can get with just two loci, each locus having two alleles. And you can kind of see right up front is here is our classic ratio, where you get the dominant A and dominant B form, 9, dominant A, recessive B, recessive A, dominant B, recessive, recessive. And so your phenotypes end up being 9 to 3 to 3 to 1. But sometimes these loci can interact in multiple ways. Dominant A can suppress B. Recessive A can suppress B. Okay, this is kind of like where we've got, right, in our example, right? The homozygous small e in coat color can suppress the B. And so as a result, when you make the cross, you don't get a 9 to 3 to 3 to 1 ratio. You actually get a 9 to 3 to 4 ratio. So conceptually, you have to be a little bit careful when you're looking at Mendelian ratios and recognize that just because it doesn't meet our 9 to 3 to 3 to 1 standard, it does not mean that there cannot be other relationships among or between the genes. So anyway, again, just an indication of how complex some of these things can be. Now, I'm going to move along here. Any questions about that? Pretty straightforward. And by the way, this table is in the text. I just like my pretty colors better. Okay. What I want to move forward is to a couple sets of problems that you can do on your own. And they are all located in that problem set 1 PDF. Some of you may have done these already. But what they really are is an opportunity to use one of the classic human Mendelian phenotypes, and that is blood types. The one that you are probably most common with is, of course, the ABO blood system. The ABO is classic blood type. These are cell surface markers, markers of self that exist on the surface of all red blood cells. And depending on the combination, it is, by the way, a single locus. It is the I locus. And so if we were to look at the definition of the genes, the A blood type is produced by ICAP-A, the B is by ICAP-B, and of course, small I is the O phenotype. Now, the relationship among and between these alleles are simply that the IA and IB are codominant, meaning that ICAP-A homozygotes have A blood types, ICAP-B homozygotes have the B phenotype, but ICAP-A, ICAP-B heterozygotes have the AB blood type, okay? So they are actually functioning as a codominant. Small I, as the small letter suggests, the lowercase letter suggests, has to be is recessive to both of IA and IB, and homozygous small I produces the O phenotype. So again, a little bit more complicated because we've got a situation with multiple alleles. A second locus you may or may not be familiar with is the Rh phenotype. This is also called the rhesus factor. And the rhesus factor is defined by a single locus with only two alleles, cap-R and small r. Cap-R is dominant to small r. Cap-R, of course, produces the rhesus factor on the surface of red blood cells. This was very important in the 30s probably through the 60s. And the problem, of course, was that if you have a mom who is sometimes called Rh negative because she does not have the rhesus factor, but her child is Rh positive. Oh my God, how could that happen? Well, duh. Whoever is the father of the child must have had, right, a cap-R. So if the child is Rh positive, well, then the child has a blood surface antigen that the mom does not have. And at times, when the blood between the two, the mom and the baby, combine, whether at birth or a little bit or during the developmental process, the mom can actually mount an antibody response to the Rh positive baby or fetus, and usually not a problem during the first pregnancy, but certainly just like getting the bee sting, the more that you are exposed, you can actually become allergic to the Rh positive. And again, from the 30s to the early 60s, this became a bit of a health problem during pregnancy. And it's actually the real reason, certainly in New York City and New Jersey and Philadelphia, not so much for Pennsylvania as a whole, but blood tests were required when you got married, because it was really to define your status at Rh for the Rh factor to prevent complications. Not so much of a problem today in today's medical world. Your doctor, if you or someone in your family gets pregnant, the OBGYNs check for this, and we have ways, there are certainly medicines that can account for this, and they can do everything from sensitize the mom to the Rh positive, just like an allergy shot, so that she does not mount an antibody response to the growing fetus, and also so there's no problem with the pregnancy. So really, but very, very important during that earlier time period. Another set of phenotypes that you may not know about are MN phenotypes. MN are defined by the letter L. I should point out that the ABO blood type was discovered by a scientist named Landauer, and it was called ABO. And eventually the genes became IA and IB, and Landauer was a little cranky that he got no credit for this, and people would forget who he was. So when he discovered also the MN phenotypes, he decided to name them LM and LN so he could name them after himself. So everybody remembers Landauer's name now. And it turns out that the M and N phenotypes, even though they are markers of self, are fully codominant, but have absolutely no, they do not mount an antibody response to one another. So they are really immaterial with regard to blood transfusions, tissue transplants, etc. They have no role at all in any of that. They're just a different type of blood type. So we have 3 loci, and you should be familiar with the inheritance of each. Why, you may ask? Well, the answer is pretty easy. What this gives us an opportunity to do is to create problem sets where we could identify, we could kind of do some reverse genetics here. And so just looking at the first problem, we've got 2 parents that we know have B blood types. And when we look at their offspring, and you'll have to bear with me and assume they have an infinite number of offspring because I'm just going to give you the proportions out the other end here. They end up with 3/4 of their offspring have B blood types and 1/4 have O blood types. And the question is, from this information, what are the phenotypes of the parents? Or I apologize, genotypes of the parents. I hate these Zoom meetings. I wish we were like all in the same room here. So what do you think?
Wait, I have a question really quick.
All right, go ahead.
So when you were talking about the blood, like in the other slide, when you said that the mom would develop an antibody like response or whatever,
Yep.
does that mean like she'll like reject the baby's blood or something?
Oh yes, and
Okay.
in usually it's not it does not occur to the detriment of either the mom or the fetus or the baby in the first pregnancy, but in subsequent pregnancies, if it happens, much like a bee sting, both the mom can start to become anaphylactic and go into anaphylactic shock, but also her antibodies will begin to agglutinate the blood of the fetus, which is bad. Okay, that's not going to produce a good outcome. So yes, absolutely. That's why it was such a huge health problem.
Sorry, I was just wondering.
No, it's a good question. So here what we're really doing is I'm asking you to think backwards, because you got to think both forward and backwards. So if you think about a Punnett square, what we really have is, in our example, and this should be, you'll be figure this out pretty quick. What do we know if there's an O blood type? What must the genotype be?
Heterozygous.
No, O. O is, right, recessive. Homozygous recessive. The O blood type has to be the small I allele. All right. What about to get a B blood type? Well, it could be BB, or it could be BB. And in fact, the way you can get 3/4 is by having this kind of an arrangement, which means what must the parents be?
Heterozygous.
Right. So this would be I cap B, small I. And so you would say that the genotypes of parents for number 1 are IBI crossed with IBI. So really, very easy, very easy. But it's to get you to think in a different direction than you're usually thinking with Punnett squares. So I'd like you to complete these for next week. I'll put up like a little Dropbox, just like we did with the other set of 4 problems, and you can submit these through the Dropbox by next Friday, okay?
Do you just want like a paper or like, I guess, like the
Some,
number and then just like what the parents are? Like we don't have to
yeah,
talk
some
about-
people, you could number them. What are the genotypes, 1 through 7, and you could fill it in. You could fill it in on the paper if you want.
Okay.
You could, some people did them by hand and took a photograph and uploaded it, and that's fine too. Let's make life easy. I don't want to make things complicated. Okay?
Gotcha.
So, Professor, is this on the first problem set
Yep.
slide?
It's the last few problems in that first slide set.
Okay, thank you.
Only problem we're doing for the for like this homework, I guess.
No, I got more slides. Hang on. These are just Punnett square practices, and these are pretty easy. I'm only going to ask you to do, just so we don't get carried away, what I really would like you to do is just this last one. So, and you could see why pretty quickly. This is practice in making gametes. So if we were to look at this first problem, you say, well, okay, what kind of gametes are we going to make? And it's pretty easy to figure out. I like to use the path method. In the old days, we never really did this, but we know at the first locus, we got I cap A and small I. The 2 possibilities for the next locus are cap bar small r, cap bar small r. And of course, then the possible gametes that this individual can produce are these. And then, of course, what I'm basically asking you to do here is to do a Punnett square. And you can see this is a very easy one, because what gametes can this individual produce?
IB and small r.
Yeah, only one kind. So not much of a Punnett square. It's kind of a Punnett rectangle or a Punnett row. So these are pretty easy. They're good for practice. But the idea is, again, just to show you how a relatively simple genetic system can result in tremendous variation in phenotypes. Of course, one of the reasons that blood typing became very important, again, when we think about it in terms of forensics, was the ability to identify parents by blood type. And so again, we're building on this idea of working backwards. So in this case, we know that there's a child with a blood type O, Rh positive, Mn, all right? And the mom is O, Rh negative, Mn. So the question is, which of these individuals, these are all the men, which of these individuals could be the dad? So in order to figure this out, what you really need to do is to figure out the genotypes of the child and the mom. So if we're figuring out genotypes, what's the genotype of the child? Well, if it's blood type O, must have a genotype of,
I.
right? Homozygous small I. But what about Rh positive? So if it's positive, it has to be a cap R, but we don't know the other one. And how about Mn? Well, M and N, if you recall from the slide, is codominant. So it must have one each of these. How about the mom? Well, the mom is type O. Rh negative, which means her genotype must be what?
Little r.
Right. And of course, Mn is the same as the child. So now, again, using our Punnett square and our knowledge of inheritance in a different way, which of these men could possibly be the dad?
Definitely has to be the first or the second one.
Well, would it be this one?
The second, maybe?
Well, wait. Let's go step by step. Can it be the first one?
No,
No.
because AB.
I
said no because the AB, but I'm not sure.
Right, because what does that genotype have to be?
IA and IB.
Right. So when it makes a gamete, it means the child, if the mom is small I, the child would have to get either the A or the B.
Right.
So we can eliminate this guy. How about this one?
That's good.
Well, again, remember,
No, because there would be a dominant IA again.
right, but what if he were heterozygous?
True.
Same thing for the R, and the M and the N are possible. So how about the next one?
No.
No, why not?
Because in order for the offspring to have a positive, Rh positive, the father would need it.
Got it. Excellent. So this one's small r. So the child can't be positive because the mom is homozygous negative, but so would this individual be. So he cannot be one of our potential dads. How about this individual here?
No.
No, why not?
It's Rh negative.
Same problem, right? The Rh negative would mean he would have to be RR and would not contribute to cap R. So it looks like our suspect is the second individual of these four. Now the question is, does this mean that this man is the father of the child?
Not necessarily.
No, why not?
Because a lot of people could have that same blood type.
Excellent. Excellent. So the point of the exercise is to show that parentage analysis can only exclude individuals as parents because it depends on the percentage of individuals in the population that have that particular set of phenotypes and or genotypes. So parentage analysis is always only exclusionary. Turns out that when you're only using blood typing alleles, it turns out that you're restricting about really about 15 to 20% of the population, again, depending on the combination of alleles. So really, there are 15 to 20% of the males that might have that particular array of genotypes. So you can't say, well, what if it was this individual's brother? What if it was a relative? What if it was just some other random person with those phenotypic characteristics? But imagine, if you will, and we will do this in the last few weeks of the semester, imagine if you knew the person's genotype at all 20,000 loci. Is parentage analysis still only exclusionary? And the answer is, well, actually, yes. It is still only exclusionary, but the difference is there are so few individuals with exactly your array of 20,000 different genes, okay? You are probably 1 in 10 to the 23rd or so individuals. Well, guess what? The population of the world is only 10 to the 9th, right? So the probability of you being the only one of that genotype is really 99.21 more nines, which is probably pretty good odds. Even I would probably bet my house that if that matched, that would be true. All right. So an interesting approach to parentage. And in fact, here are some parents. And again, this is an exercise that I'd like you to do in the problem set. Do this for next week. This shows you that a woman is married to one man and produces 4. By the way, they are legitimate children. 1 year I gave this, I said she has 4 children, and a student said, well, you don't know if any of them are really from another man. Maybe the postman rang twice. And so I changed it to say legitimate. These are really hers, okay, and only hers with that individual. What are the genotypes? And I'm asking you to predict the genotypes of the parents. So pretty straightforward. And that leads us right to the end. And perfect timing. So real easy, I think. I don't think you'll have much problem with the problems. There will probably be 1 or 2 of these on the exam next Friday. Okay, the exam will be on Blackboard. So as a result, it will be available starting Friday in the morning. So it'll be available starting, I'll probably make it like 8 a.m. or something. And then you'll have the entire weekend. So it will be available for you to take between Friday, 8 a.m. and Sunday, whatever it is, 11:59 p.m. Okay. So.
No, we don't have to show up to class or anything, right?
Pardon me?
We don't have to show up to class or like show you that we're like here or whatever.
No. I'm going to assume we're still grown-ups, and we are not going to have a Zoom meeting for next week, and I'll send a reminder out about this. First of all, you've got the exam to do, so I don't want to give you more information to complicate matters. Second, I have a really exciting event to attend on Zoom, unfortunately. But one of my research students from Stockton is defending her dissertation in genetics at Penn State. And so I'm very excited. She was, most of you, well, unless you knew her from before, one of her sisters, Alexis Sullivan is the student. She was one of my research students, worked with me for 2 years here at Stockton on some really 2 different projects. And she wanted to go to grad school, got into the genetics program at Penn State, and is graduating with a degree in genetics and genomics at Penn State, doing some really cool work on full genome sequencing of lemurs and the relationship among different populations of lemurs to the human genetic map. So really, I'm really excited for her. It's great to see her do so well. And so I'm going to attend her presentation by Zoom, which is at 9 o'clock next Friday. So I'm going to try to get her to make a recording of this, but the committee has to agree. And if they do, I'll make it available to you guys, for those of you who may be thinking about grad school or a career in research, just to get some idea of what happens to you during the process. I'm super proud of Alexis. She just did a great job. If you want to see one of her early projects, I believe her poster on the Sordaria is still in the presentation board outside of the genetics lab in USC1. And she was the only one actually who could consistently get great spreads to look at the ASCII. And so we did some genotyping and some genetic linkage experiments with the Sordaria with her, and she did great work. Later on, she did even better work doing some other biochemical stuff we were working on. And I'm really proud of her. She did, and it's going to be great to see her graduate. And especially from a place, some of you know, I used to be head of genetics at Penn State, so it'll be great to go back and see her tidy up her degree there. So I'm excited. Sorry I won't be here for you guys, but we'll see if we can make something work.
Professor Barbata, I have a question.
Yeah.
So, for the last slide that you just showed us with the legitimate children.
Mm-hmm.
So for the first one, when it says both of them are like II, which would mean like O blood type, how do you know that her parents could be like heterozygous for like A or B, or both of them could just be O? Like, how would you know? Like,
well, you may not. So you kind of, and by the way, as I recall, you may not, you know that they both have to have, because these are legitimate children. If you look at the slide, I don't know, can you guys see this? Wait a minute.
well, I mean, I have it in front of me, so like
Yeah, but everybody else doesn't. So hold on one sec. Doodly-doo. So this is just not in full screen mode, but I'm assuming you guys can see this.
yeah.
So if we were to look here, let me see if I can get my pen working. Oh, maybe not. Oh, yeah, okay. So, for example, if you were looking at just II, you know, because these individuals are homozygous recessive, you know that parent 1 and parent 2, notice I'm not indicating male or female. You know that they both have to have small I, right?
Yeah.
And one has got to have A and the other one has to have B. We don't know in the nature of this problem which is which.
Right.
But we know that one or the other does have to have that.
But both of them could also be homozygous for I's too, right? So like both of them could just both have blood.
Well, then how do you get a B and an A?
Oh, I was just talking about the first one.
Yeah, I am. Oh, the first individual, but these are all 4 children.
Oh, hello, duh. Okay, gotcha. That makes more sense now.
Okay?
Yeah. And then another question is, so from the
Mm-hmm.
last problem set, my question, sorry, my phone's going off. My
Oh, good. I'm
question
glad it
was,
was you. I was looking at all my devices here
Right.
thinking, oh my God, we're dinging.
sorry, my phone keeps affecting me. God. So
All right.
how would you know from the F1, like, what combination of alleles will make the F2? If that makes sense.
Well, an F1 generation is always heterozygous.
But what if you knew that the parents, like one of them was heterozygous and the other one was homozygous, and then they make F1 and like they can make all kinds of different
Oh, yeah, I see what you're asking. No, you would just have to make all, wait a minute.
because I kind of just included all combinations because I didn't know which one to choose from in the problem set. And I
Yeah,
didn't
no,
know-
there is no choosing.
Oh, okay, so you would just include all of them.
Yeah, exactly.
Okay, I just didn't want a chance getting it wrong, so I was like, how do you know which combination is gonna
No, take a chance. It's all
together.
good.
Okay, I just didn't want to like get it wrong when I like knew that there was other combinations that could occur.
Yeah, no, it really is a function of assuming that all possible combinations exist in the next generation.
Okay, so then you would just list all of them depending on
Yep.
which,
And as I mentioned, I think I mentioned in the, perhaps it was just our emails or perhaps on Piazza, there are multiple ways to solve that problem. The most time consuming, of course, is just making however many Punnett squares. So they're, and then counting everybody up. So you
Right.
could do that. It's a correct answer. It's kind of like our probability problems is you can count them,
mm-hmm.
but counting them is not always the most elegant solution.
Gotcha.
So there are other ways to do it, but just doing those Punnett squares would be correct.
Okay, cool.
And the reason I don't differentiate, by the way, is because strangely, even though you might do the calculations using, for example, an algebraic or a path diagram, if you were to program a computer to answer the same question, you would basically have to do it the longhand, make all possible combinations and count them. So I don't distinguish between the two because there comes a point in time if you proceed a little bit further down the line, it'll be easier for you to get a computer to program it to do it the long-handed way, which of course having the computer do it means it solves it in milliseconds, as opposed to trying to get it to do the logical way, which is a completely different kind of programming and probably far beyond where we'll ever be as undergraduates. So, not everybody.
Factorial stuff, right?
What's that?
You're talking about the factorial stuff?
Oh, yeah,
Okay,
definitely.
gotcha.
Definitely.
Okay. All right, that was all my questions, I'm pretty sure.
Cool.
Thank you.
All right, everybody. We good? Anybody else?
So with the problem,
Professor,
Go ahead. It's all right.
the
you
problem,
go first.
okay, with the problem sets, we're leaving off from where we last left off to the last slide that you showed us today.
Yes.
Okay.
Yep. They're just I think they're the last 4 or 5 slides.
Okay. Thank you.
Yep. Anytime.
Professor,
Yes.
I had a question about the Piazza. I think I did post a question. You said you would be uploading the playlist shortly. You did not upload it yet, right? Or did I?
What's that?
That playlist?
Oh, no, I'm sorry. I'm a little bit behind on my Spotify,
Okay.
but it'll be up shortly.
Okay, I'm just making sure.
No problem.
Okay, thank you. Have a good day.
All
Actually,
right, everybody have a
another
great week.
question. I have another question, sorry.
Okay.
The one slide, you said you only want us to do the combination of the one all the way at the bottom, right?
Yep, the last one.
Okay, cool. All right, that was it.
And you'll know why once you start it.
Okay.
All right, everybody have a great weekend. As soon as this is done collating, I will post this for anybody who missed it or if you want to go back and revisit some of the commentary. Okay?
Thank you. Have a
Professor,
good weekend.
Take
Bye.
care, you guys. Thanks.
Have a good
Be
weekend.
careful out there.
Have a good weekend, Professor.
You too.
I was able to
Bye-bye.
do the quiz for the reading, but I
Oh,
wasn't
yeah.
actually able to do the assessment because it's time locked.
Oh, shoot. I'll go try to release it, but if I can't, don't worry about it.
Okay, thank you. I
Alright, no
I
problem.
was looking for things. I was like, oh, I know we probably have something, and it just clicked to me this morning, like, hey, I should probably check Sapling Plus.
It happens. No worries.
Thank you.
All right. Take care, Ryan.
You too.
All right. We'll see y'all.